So we will be talking about the cell organelle endoplasmic reticulum today. During the lecture, we'll talk about the structure of endoplasmic reticulum, its types, and its important functions in eukaryotic cells. So before formally start with the lecture, I'll just tell you a few very special things about endoplasmic reticulum. Now, endoplasmic reticulum. Once we picture this cell organelle in our mind, or once we start talking about this cell organelle, sometimes I've seen that with many students, they actually do not know its very important role in the cell. Now, once we had talked of the role of different cell organelles in the previous lectures in general, I had told you that each and every cell organelle has a specific function. Now, this endoplasmic reticulum, once you'll learn more about this, you'll understand also. This particular cell organelle, you'll see it performs most of its vital functions in coordination with other cell organelles, like the nucleus and the Golgi apparatus. So, once I'll teach you this, just keep this thing in mind and try to understand its important functions in keeping this particular thing in mind, right? So, I'll just begin with endoplasmic reticulum. So, endoplasmic reticulum is a network of interconnected internal membranes. Generally, it is called the largest membrane in a eukaryotic cell, an extensive network of closed, flattened membrane-bound sacs called cisternae. I'll just show you a figure, a diagram of the endoplasmic reticulum. Here you can see, which will quickly just reflect the structure of endoplasmic reticulum to you. Now here you can see the yellow part here. Okay, it represents the cytosol of a particular cell, and you can see the cell membrane being drawn here. And this is the nucleus, and in continuation to the nucleus, you can see the endoplasmic reticulum. Okay, now sometimes it is shown in diagrams, it is shown dissociated from the nucleus, and sometimes it is shown in continuation to the nuclear membrane. The reason being that it is said that if we talk of the biogenesis of the endoplasmic reticulum, it is said that it has actually originated from the outer nuclear envelope only. Okay, and its membrane reflects a number of structural similarities and composition of its biochemical constituents which are similar to that of the nuclear membrane. Okay, so that is why we always see the endoplasmic reticulum drawn in continuation to the nuclear membrane. Okay, and then of course we'll come back to the figure once we'll talk of many important things. So the term or the name endoplasmic reticulum was coined by Potter in the year 1953, who had observed these cell organelles in the electron micrograph pictures of liver cells. Endoplasmic reticulum has a number of functions in the cell, but it is particularly important in synthesis of lipids, membrane proteins, and secreted proteins. Now, you must not get a false notion that endoplasmic reticulum actually synthesizes proteins, right? Once we talk of its significance in synthesis of membrane and secreted proteins, you should never be confused. The synthesis of proteins is always performed by the ribosome, right? Now, that is why in the beginning of the lecture, I have told you that you'll see that most of the important functions of the endoplasmic reticulum are performed in association with the other cell organelles. Now, I'll again just go back to the figure. You can see here that some part of the endoplasmic reticulum is containing ribosomes attached on its surface. You can see these small granules that are shown on this in this figure, which are attached to the surface of the endoplasmic reticulum. And here you can see this part is called the rough endoplasmic reticulum, because under the microscope it appears to be granular, and its surface gives a rough appearance. It is all because of the presence of these ribosomes. And here you can see this part, which is called the smooth endoplasmic reticulum. It is not containing any ribosomes. So basically, when we say that it plays an important role in the synthesis of proteins, so it is not the endoplasmic reticulum that is actually synthesizing the proteins. It is these ribosomes which reside on the surface of the endoplasmic reticulum, that is the rough endoplasmic reticulum. They synthesize proteins on its surface. Now, there are other ribosomes also which are present free in the cytosol. Once we'll move to ahead in your syllabus to the part where we will talk of protein sorting and vesicular trafficking in eukaryotic cells, there you'll see that all the proteins that are synthesized by a cell, right, they are utilized in different compartments of the cell, and the process of sieving or sorting proteins involves certain proteins that follow a specific pathway through the rough endoplasmic reticulum. They are specially directed to some special cell organelles, right? And the ribosomes which bear mRNA for those proteins, they actually go and reside on the surface of this endoplasmic reticulum, making it look rough or appear granular. So this is its role in the synthesis of proteins. We'll also talk there about the mechanism of the synthesis of secretory and membrane proteins and how they are sorted on the surface of rough endoplasmic reticulum itself. But for now, you should know that how it plays a role in protein synthesis. Okay, so it is not actually synthesizing proteins. I'll make it clear again. It is only providing a surface for the synthesis of proteins. And the smooth part, okay, which is not granular, this smooth endoplasmic reticulum is mainly involved in the synthesis of lipids. So I hope this is clear to you. So I'll just again read this sentence for you. The endoplasmic reticulum has a number of functions. I mean, it has many other important functions than participating in protein synthesis and actually synthesizing lipids. You'll see ahead that it plays a very important role in protein sorting and also in proper folding of proteins to align these proteins in their correct three-dimensional state, and adding carbohydrates to proteins, that is the process of glycosylation, that also starts in the endoplasmic reticulum. So these are other functions you'll see which are performed by the endoplasmic reticulum, but it is particularly involved in synthesis of lipids, membrane proteins and secreted proteins. The occurrence of ER, that is the endoplasmic reticulum, is in eukaryotic cells, and their location actually varies from cell to cell. Their number also varies from cell to cell, that you can see is mentioned in the next point. The erythrocytes, that is the RBCs, egg and embryonic cells lack endoplasmic reticulum. Now, their number per plant cell can vary from several hundreds, as in tissues of corn root and algal rhizoids, to a single organelle in some algae. Actually, you'll see that different type of cells, different type of eukaryotic cells in any living system, right? They have different requirements for protein synthesis. The extent to which they synthesize proteins, I mean how active or how less active they are, it matters, right? In terms of the occurrence of the endoplasmic reticulum, to which part of the cell they are going to be located, I mean what is going to be their position and especially their number. So they are seen more in case of cells which readily or actively synthesize proteins time to time. So again, take a look at this picture. You can see here the nucleus, nuclear pores. We have talked about them in the previous lecture. And then you can see the rough endoplasmic reticulum being covered by the ribosomes, the smooth part. And here you can see that how it is actually transporting proteins, forming vesicles to the next cell organelle, that is the Golgi apparatus. Of course, after endoplasmic reticulum, we'll be talking about the Golgi apparatus, and then you will see how both these organelles are actually associated in terms of glycosylation, that is addition of carbohydrates to proteins. We've seen that the best example would be the membrane proteins. We have seen the glycocalyx. Okay, if you remember the structure of the plasma membrane, you must remember that the outer face of the plasma membrane is rich in glycoproteins and glycolipids. So this process of addition of carbohydrates to proteins, it is actually completed by the ER and Golgi apparatus together. So again, this also is a very important role of endoplasmic reticulum in eukaryotic cells. And finally, you will see that together with the Golgi apparatus, then it is involved in sorting these proteins, folding these proteins, adding carbohydrates, these proteins, and allowing these proteins to finally move to the plasma membrane. Then we must talk of the morphology of the endoplasmic reticulum. Now, if you take a look at its morphological features, you'll see that it occurs in three forms, right? You can see three distinct forms in the endoplasmic reticulum. First one is the lamellar form or the cisterna, which is a closed fluid-filled sac, vesicle or cavity. This is called the cisterna. Then we have the vesicular form or the vesicle and the tubular form or the tubules. So technically it is divided into cisternae, vesicles, and then the tubules. So the cisternae are long flattened sac-like unbranched tubules having diameter approximately 40 to 50 micrometer, and they remain arranged parallelly in bundles or stakes, and this part is actually covered mostly by the rough endoplasmic reticulum. Okay, so here you can see the cisterna. Then it has the vesicles. The vesicles, as the name defines, these are small oval membrane-bound vacuolar structures with a diameter ranging from 25 to 500 micrometer, depending upon the size of the vesicle and the cargo it is carrying. And they often remain isolated in the cytoplasm and occur in most cells, but they are specially abundant in the smooth endoplasmic reticulum part. Okay, here you can see the vesicles, and this one of the transport vesicles, you can see it is a part of this endoplasmic reticulum structure only. It arises from the endoplasmic reticulum only. You must have seen that whenever we see a picture or we draw a diagram of either endoplasmic reticulum or the Golgi apparatus, we always draw the very small vesicles towards the trans side of ER. Okay, now I must also talk to you about the cis and the trans phase of ER. The part of ER that is actually facing the nucleus, it is called the cis phase, and the one that is actually facing the Golgi apparatus, it is called the trans phase. Similarly, the part of the Golgi apparatus that is facing the endoplasmic reticulum is called the cis phase, and one that is towards the cell periphery, that is called the trans phase. Okay, so you'll see that the cisternae are rich towards the cis phase of endoplasmic reticulum, where it is connected to the nucleus, which is facing the nucleus, and the part which is actively involved in synthesis of ribosomes, because their structure provides a very large surface area. Okay, these foldings, they provide large surface area on which number of ribosomes can reside, and that protein synthesis can actually take place. Now, towards the trans side, you'll see that all the proteins that have, after synthesis by the ribosomes present on the rough endoplasmic reticulum, they'll enter into the ER and they'll move to the medial and then the trans phase, from where they will be then again move to the Golgi apparatus for final modifications, right? So you'll always see that those proteins will get packaged in small membrane bound vesicles that are going to arise from the trans, you can say, ER network. That is why the trans side, which is actually facing the Golgi apparatus, is rich in all, or you can see it contains a number of these vesicles, because all these vesicles are actually carrying all the proteins that will be then transported to Golgi apparatus for final modifications like folding and the glycosylation. So, the last part, that is the tubules, are branch structures forming the reticular system along with cisternae and vesicles. They actually connect these, and they usually have the diameter of 50 to 190 micrometer and occur almost in all the cells. Tubular form of ER is again found in the smooth endoplasmic reticulum and is dynamic in nature. That is, it is associated with membrane movements, fission and fusion between membranes of cytocavity network. It is actually this tubular part only that give rise to these vesicles that can, allow the fusion and fission, that is formation of vesicles from the endoplasmic reticular membrane. Then let's talk of the types of endoplasmic reticulum. You all must be knowing that endoplasmic reticulum appears in two types, rough and smooth. So the agranular or the smooth endoplasmic reticulum is the part of the ER with no studded ribosomes that makes it smooth in appearance. The adipose tissues, brown fat cells, and adrenocortical cells, interstitial cells of testes, and cells of corpus luteum of ovaries, sebaceous cells, and retinal pigment cells contain only smooth endoplasmic reticulum. The synthesis of fatty acids and phospholipids takes place in the smooth ER. It is also abundant in the hepatocytes. Okay, so the smooth ER is mainly involved in the lipid synthesis, and it is actually the part of the endoplasmic reticulum that is devoid of the ribosomes, that is, it is not involved in protein synthesis. Then the next type is the granular or the rough endoplasmic reticulum, also called the RER, because it has ribosomes bound on its surface that makes its appearance rough. The rough ER synthesizes or helps in the synthesis of certain membrane and organelle proteins and virtually all proteins that are to be secreted from the cell. Please excuse me. Now, uh, all the proteins that a cell synthesizes that are actually going to be secreted out of the cell, all those proteins are actually synthesized by the ribosomes that are present on the surface of the rough endoplasmic reticulum. The cells of those organs which are actively engaged in synthesis of proteins, such as acinar cells of pancreas, plasma cells, goblet cells, and cells of some endocrine glands, are rich and contain rough endoplasmic reticulum, which is highly developed in them. Then we must talk of the functions of the smooth endoplasmic reticulum and the rough endoplasmic reticulum, the common functions and the differences that we have already talked of. So the endoplasmic reticulum provides an ultrastructural skeletal framework to the cell and gives mechanical support to the colloidal cytoplasmic matrix. This you can easily judge by again just taking a look at the diagram that I had shown you in one of the previous slides, where you will see how extensively it forms a skeletal framework that provides mechanical support to the cytoplasmic matrix. Then the exchange of molecules by the process of osmosis, diffusion, and active transport occurs through the membranes of endoplasmic reticulum. And for this purpose, the ER has various permeases and carriers. You can see that all these important processes which are performed by the plasma membrane, very common processes that you must have heard of, you can see all these processes also occurring in the endoplasmic reticular membrane also. Once you move ahead in this syllabus, you will learn a number of molecular processes where you will see how these important membrane transport processes help ER regulate a number of cytoplasmic functions. The endoplasmic membranes contain many enzymes which perform various synthetic and metabolic activities and provides increased surface area for various enzymatic activities reactions. Like, it is because of its folded structure, and you can see that increases the surface area for all these particular activities. Then the endoplasmic reticulum also acts as an intracellular circulatory or transporting system. Again, we'll talk about this in detail once we'll talk about vesicular trafficking in eukaryotic cells, where you'll see how this sieving and transportation of membrane and organelle proteins and lipids are performed by the endoplasmic reticulum in association with the Golgi apparatus. So it definitely acts as an intracellular circulatory and transporting system. The ER membranes are found to conduct intracellular impulses. For example, the sarcoplasmic reticulum, okay, that is found in the muscle cells, transmits impulses from the surface membrane into the deep region of the muscle fibers. The sarcoplasmic reticulum plays a role in releasing calcium when the muscle is stimulated and actively transporting calcium back into the SR when the stimulation stops and the muscles must be relaxed. Now, you know, muscle contraction is associated with the rise in cytosolic calcium concentration, right? So endoplasmic reticulum, apart from all its functions in transport and protein synthesis, it also acts as intracellular reserve of calcium ions, right? So this means that apart from the calcium channels that are present on the plasma membrane, the cell can also, raise the cytosolic calcium levels through release of calcium ions from the endoplasmic reticulum. That is, in case of muscle cells, that is the sarcoplasmic reticulum, when stimulated through a proper response. And again, the excess of calcium can be stored in the sarcoplasmic reticulum also for future use whenever it is required by the cells and the extracellular sources of calcium are not available. Okay, so this is all about endoplasmic reticulum that we have to talk of. Thank you very much.